So we have seen that how the catalysis of long chain fatty acids which are even numbered, which have even number of carbon atoms, it takes place. But keep in mind that not all the fatty acids are even numbered. There are certainly some odd chain fatty acids also, and for odd chain fatty acids there is a little bit of what's say alteration in the process of beta oxidation, although I mean almost all the steps are the same. Only one step in which you get the final product, it is not the acetyl CoA, but it is the what we say the propenyl CoA. So all the four steps we have been talking about in the beta oxidation of long chain fatty acids of the even number carbon atom containing compounds, that remains there. But when you get the final molecule, that is not acetyl CoA, it will be propenyl CoA. Now the propenyl CoA cannot be converted into the acetyl CoA in the same through the same enzymes which are used when we are dealing with the beta oxidation of the even number of compounds which contain even number of carbon atoms. Here the enzyme machinery have to be a bit different. That's why the nature have devised a different way of dealing with this. So as I have told you, at the end of this chain of events which are involving the oxidation of the odd chain compounds, what will happen that you get the propenyl acid, this propenyl CoA. This propenyl CoA then is converted into a methyl mannoyl CoA through the activity of a carboxylase enzyme. This methyl mannoyl CoA is converted into L-methyl mannoyl CoA through the activity of racemase enzyme. This racemase enzyme then what will it do, that it will change the configuration of this compound. First of all, the dimethyl mannoyl CoA you get from the carboxylase activity, and then through the activity of the racemase enzyme you get the L-methyl mannoyl CoA. So only the configuration is changed. Once you get the L-methyl mannoyl CoA, then you get the final product of the this beta oxidation, that is the succinyl CoA through the activity of the mutase enzyme. So L-methyl malonyl CoA is acted upon by the mutase enzyme, which act with the help or with the support of a coenzyme form of vitamin B12, and you get the succinyl CoA. Once you get the succinyl CoA, and of course it will become part of the Krebs cycle and it will undergo the further degradation. Then there is another exception to the traditional way of beta oxidation when we are talking about the beta oxidation process, is that you not all the time get because when we were talking about the oxidation of the palmitic acid, it is a saturated compound, but it's not all the time you are dealing with a saturated compound. You sometimes also deal with those fatty acids which have unsaturation, which have a double bond in them. So for these unsaturated compounds, nature have devised a different way. Why nature have to devise a different way? Because these intermediates of these double bond containing compounds, they cannot be acted upon by the hydratase enzyme. So what happened here is that in case the double bond is at a carbon atom which is for instance which is odd number, let's say a carbon atom number 18 into 1. So what will happen then? You require the 3,2-enonyl CoA isomerase. That is if the double bond is at an odd number carbon atom. But if the double bond is at an even number atom, carbon atom, that's in the case of double bond is in the form of 18-2 configuration, 18-2, that is double bond is at carbon atom number 2, then it will require, in addition to the isomerase enzyme, it will require dienolyl CoA reductase enzyme. So this NADH2 dependent enzyme has to be needed in addition to the isomerase enzyme if we are talking about the even number, if the double bond is at the even number. So it's not odd, read it even number carbon double bonds. Finally, one final exception to the traditional or conventional oxidation, beta oxidation pathways is that usually beta oxidation is for less than 20 carbon atom long fatty acid compounds. But what if it's even longer? That's another very long chain fatty acids. These very long chain fatty acids cannot be oxidized into mitochondria because mitochondria don't have that machinery to deal with them. So for that, they have to be moved toward in the peroxisomes, and in the peroxisome, then they are acted upon by enzyme, which then divide these long chain fatty acids into those molecules which are relatively shorter chain, or at least in the range which can be acted upon by the enzymes of the mitochondria. So these relatively shorter chain fatty acids then will move towards the mitochondria, and then they will be further oxidized by the traditional way of the beta oxidation. So now we ask certain questions as we have finished very important part of the lipid metabolism, that why fatty acids are so highly energy yielding. We have showed you that if you are dealing with 16 carbon fatty acid compound, it will give you 129 molecules of ATP, which is way more than what you get from the Krebs cycle of or from glycolysis when we were dealing with the metabolism of glucose. So what is so special in the fatty acids that they give so much energy? If they are giving so much energy, then why not lipids are the, or specifically the fatty acids, are the major source of energy? Why it's most of the time glucose which act as the major source of the energy? Of course, fatty acids, we already know that they are the source of the energy. Magar kya wajah hai that they are, but what is the reason that they are not as main a source of energy as is the glucose? Then why it's more all the time beta oxidation? Why we don't talk much about the alpha oxidation or gamma oxidation? What is so special about the beta oxidation? Finally, why, I mean, it's the easiest one, by the way, why this oxidation process takes place in the mitochondria? Why not in the cytoplasm? Think about it. Let's see to what extent you can dig deeper. Thank you.